Hello dear students, this is the last part of Chemistry 280, Nucleic Acids. In the nucleic acids, we have to begin by the building blocks. The building blocks of nucleic acids are nucleotides. They are made of three different units. A phosphate group, as you can see here, a sugar, that is a pentose because it contains 5 atoms of carbon, and a nitrogen base that could be a purine or a pyrimidine. Here we have the different nitrogen bases. The pyrimidines can be, as you can see here, the pyrimidines can be the cytosine, the thiamine, and the uracil. And the purines could be adenine and guanine. In the ribose, the carbons are has a different number. That's why it's important for us to recognize the carbon 3 and the carbon 5. You know that in the sugars in RNA is the ribose, but in DNA is the deoxyribose. The different types of nucleotides has the different combination of deoxyribose or ribose with the different nitrogen bases in order to make DNA. Remember that in DNA we have adenine, cytosine, guanine, and thiamine, while in RNA we have adenine, cytosine, guanine, and instead of thiamine we have uracil. The different nucleotides make a polymer by joining. The phosphate group is the joining of the different sugars, and the sugars are joined to a nitrogen base, as we have seen before. But it's important for us to understand that the ending of DNA could be 5 or could be 3. The same thing for RNA. The difference between them is in the type of sugar, as we can see, deoxyribose in DNA and ribose in RNA, and also that we have uracil in RNA while we have thiamine in DNA. The nucleotides could be present in the energy carriers. For example, we have heard about ATP. What is exactly ATP? Adenosine triphosphate. What does it mean? That we have 3 phosphate groups. In ADP, adenine diphosphate, we have 2 phosphate groups. In adenosine monophosphate, of course, we have only 1 phosphate group. They are used in the reactions in the cells to produce energy. These two guys that you can see here, Crick and Watson, were responsible to predict the structure of DNA. The Chargas rule established that adenine only can be combined with thiamine and guanine with cytosine. Remember, A with T, G with Z. Thus, by X-ray diffractions, these two guys that you can see here, Rosalind Franklin and Maurice Wilkins, establish the periodicity of the different structures in DNA. Here we can see how the adenine only combines with thymine in DNA to make a double strand structure, while guanine combines with cytosine, as you can see here, making hydrogen bonds and making this double strand structure characteristic of the DNA. But look at this. This strand begins in 5 and ends in 3, but the parallel one is opposite, begins in 5, begins in 3 and ends in 5. The double helix is separated at 3.4 angstrom from one to another. That's the distance of the nitrogen bases joined by hydrogen bonds. And you can see here the different dimension and a schematic of how it is represented. Remember that DNA has a double strand structure, while RNA has only a single strand structure. One of the most important processes in DNA is replication. In order to have an idea of what is replication and the importance that it has for the life, we are going to watch this video.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Okay, now we have seen the process of replication. Replication is so important because it's a way that we can produce more units of DNA. DNA contains the genetic information for all the different processes in our body. Not only because it's for having the color of the eyes of the mother or the father, also it has the information for the production of every single protein in our body. And as I told you before, a protein can be anything. In our body, they could be enzymes, and there is not a process without enzyme action. They can be a hormone that regulates the different processes in our bodies. It could be defense, it could be anything in our bodies. That's why DNA is so important in the production of proteins. Let's see this part that is very important. How to go from DNA to a protein. In this process, or I will say in this process, but it's a very complicated one that has different steps, we are going to see transcription and translation. And for that, we are going to see this video.
In order for our bodies to function, we need to supply them with a variety of nutrients we get from our diet. Our bodies cannot use the food as it is when it enters our digestive system. The process of chemical digestion uses different proteins and enzymes to break down the food particles into usable nutrients our cells can absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes, transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in three stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where